Okay, so pyruvate in the cytoplasm, the cytosol, has been transported into the mitochondria. In the process, one of the carbons off of the 3-carbon chain have been cut off, so you end up with a 2-carbon chain that has an coenzyme A attached to it. What we see over here is a series of reactions. Well, they call it a cycle and not a pathway. Pathway has a beginning and an end. Beginning, end. What you see here is a constant churning. Okay? It's churning inside the mitochondria. There's no beginning, there's no end. It's just a constant churn. Stuff gets added in, stuff comes out, but it's a still constant churning. No beginning, no ending. So what do we see when we look at this? Well, first off, what we see here is here's our incoming acetyl-CoA. Our acetyl-CoA will be added in to form citrate, hence the citric acid cycle. Citrate enzymatic reaction gives you isocitrate, alpha-ketoglutarate, succinyl-CoA, blah, 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 on and on and on. Notice the difference between some of these molecules is not that great. Here you see more NADHs are made, but more importantly, boom, right there, a CO2 is given off. Another of the carbon dioxides you breathe out. C6, 6, 5, 4, there to go. The other two CO2s that came in were the second carbon dioxide that came in as acetyl-CoA. But now you have a C4, succinyl-CoA. Succinyl-CoA can't do anything with, okay? Can't end here. Somehow you have to get succinyl-CoA and turn it back into oxaloacetate. That's what the second half of the citric acid cycle is all about. Rearranging things, stripping off hydrogens, adding stuff back in, hydroxyls back in, so that succinyl-CoA can look like oxaloacetate, which can now accept a new acetyl-CoA. Remember, shape dictates function. The shape of succinyl-CoA is really nothing like oxaloacetate. So the enzyme that takes acetyl-CoA and adds it to make citrate will not recognize succinyl-CoA. So you got to do some rearranging. That's what these steps are all about. Here's the thing though. The carbons that come in right here are not the ones that are going to be released right there. No. What we see happens is these 2 carbons that came in are going to have to spend some time going through this process. Those carbons, the first one to let go is right there. Second one to let go is right there. They have gone through the cycle twice. They come in, go through the cycle once, go through the second, and on the second time around, going into the third, boom, boom. That's when they get let go. So, incoming acetyl-CoA has to go through twice before it's released as carbon dioxide. This is the way we've evolved so that there's constantly the churning mass of enzymes and substrates so that the citric acid cycle never begins, never truly ends. It's always there. So you look a little bit closer. You see CO2, NADH, NADH, CO2, and GTP. GTP, kissing cousin of ATP, does roughly the same thing. It's just not found in as great as concentration in our cells. FADH2, it does the same as NADH. It escorts an excited electron to phase 3, oxidative phosphorylation. So far, up to this point, for every 1 glucose, get 4 ATP, 2 from glycolysis, 2 from citric acid cycle, and you end up with 6 carbon dioxides. All the carbons from glucose have been completely consumed and released as a gas. What was a solid, a sugar, a solid, has now been released as a gas, CO2. You've also got 10 ADHs, so you got 10 excited electrons, not a lot, but it'll be enough, and 2 FADH2s. And we'll see what happens in the next.